Good afternoon, everyone. I'm Verdiana Di Giulio, a first-year PhD student in cellular and molecular biotechnologies at University of Teramo. My research project is about discovering new insights on hybrid cells to improve tissue regeneration. Regenerative medicine is a scientific field which aims to replace tissues or organs that have been damaged by different causes, such as age, disease, traumas, or congenital issues. Current treatments are mainly distinguished into three categories: molecular therapy, which act on RNA or genes; tissue engineering, which is about the construction of artificial organoids or scaffold in a laboratory that have then to be transplanted into the damaged site; and then we have cell-based therapy. The cell-based therapy promotes the repair of the diseased, dysfunctional, or injured tissue using staminal cells or their derivatives. This therapy refers to the transfer of new cells through injection into the damaged site to promote healing or regeneration. Current treatments of cell-based therapy are used for corneal and retinal disorders, but also for the treatment of osteoarthritis. So, what is a staminal cell? A staminal cell is a single cell that can both replicate itself, so it has the self-renewal capability, but it can also differentiate into many cell types. Due to this intrinsic power, stem cells are used for regenerative purposes. There are so many kinds of staminal cells. In our laboratory, we work with amniotic epithelial cells, which derives from the epithelial layer of the amniotic membrane of the sheep, as we work with this animal model. Amniotic epithelial cells display different characteristics. For example, they display high stemness characteristic, high proliferation rate, that is to say that they grow really faster, and differentiation, so they can differentiate into many cell types of different parts of the body. So the heroes of my project are hybrid cells. Amniotic epithelial cells undergo spontaneously through a process called epithelial to mesenchymal transition, a process which generates an array of hybrid cells which demonstrate different characteristics. Firstly, they have mixed features, so they have features of amniotic cells and mesenchymal cells. And furthermore, they also have enhanced properties in comparison to other staminal cells, for example, immunomodulatory and migratory capabilities that are essential for tissue regeneration. So why is regenerative medicine so important? Think about tendon disorders. Adult tendons often respond poorly to current treatments because tendon is characterized by limited vascularization, low metabolism, and low cellularity. All three elements that are essential to ensure an effective tissue regeneration. Always referring to tendinopathies, due to the increase of human population ages and life expectancy, it is estimated that the 25% of the adult population suffer of a tendon-related condition. The same happens for the equine patients. In fact, the 46% of the resources will suffer a tendon-related condition that will negatively impact that kind of industry, which is valued at 400 billion euros worldwide. Tendinopathies fully represent what is the One Health approach. It is a concept that recognizes that the health for humans, wild and domestic animals, and the environment are strictly connected and linked. So what is the main goal of my research project? My aim is to characterize these hybrid cells, to use them as eventually in future as an innovative type of staminal cells which can guarantee an optimal regeneration. So the different analysis of my research project will be conducted into two steps. Firstly, in vitro, so to characterize these cells, so to discover the intrinsic features that can be exploited then in a regenerative context. And then in vivo, so with functional assays that assess the underwent tissue regeneration due to the help of our hybrid cells that are being injected into the damaged site to restore and promote a faster regeneration. So, my research project have so many beneficial and positive implications on our future society. Firstly, the study will help to depict a more accurate overview of the hybrid cells and their contribution in both physiological and pathological processes. Furthermore, as most of the research have been conducted on cancer models, this study help in the understanding of the physiological behavior of hybrid cells, thus highlighting how these hybrid cells can be useful for regenerative purposes. For the most of you who are interested and curious about this fascinating world of regenerative medicine, you can contact me on LinkedIn or by email. Thank you for your attention.